Translation is the process of synthesizing a protein from a strand of mRNA. This process involves ribosomes, mRNA, and tRNA, initiation and release factors, amino-acyl tRNA synthetases, and of course, amino acids. Translation takes place in 3 stages: initiation, elongation, and termination. During initiation, the small ribosomal subunit attaches to the mRNA along with different initiation factors. This is known as the initiation complex. The initiation complex moves along the mRNA strand until it encounters the AUG start codon, where the initiation factors are released and the large ribosomal subunit attaches. This begins the second stage of translation, which is elongation. In elongation, mRNA amino acids are added to the growing polypeptide chain of the soon-to-be protein. tRNA carries the amino acids that are specified for the corresponding codon, which was found on the mRNA. A codon is the 3 adjacent RNA nucleotides found on mRNA. The complementary 3 nucleotides found on a tRNA molecule is known as an anticodon. tRNA knows what amino acid to carry to the mRNA because of an enzyme known as amino-acyl tRNA synthetases, which adds the correct amino acid to the tRNA. You can see that the large ribosomal subunit has 3 sections: the E or exit site, the P or peptidyl site, and the A or amino-acyl site. tRNA enters the subunit through the A site. When another tRNA is added, the first tRNA moves over to the P site, where its amino acid is added to that of the tRNA on the A site. Once a third tRNA molecule enters, the first tRNA exits through the P site, and the process continues until the ribosome reaches a stop codon, in this case, UUG. This begins termination, where the amino acid chain is released by a release factor, and the ribosome disassociates with the mRNA chain. During translation, prokaryotes have a unique ability that allows for one mRNA chain to code for multiple proteins at a time. They also possess the ability to perform transcription and translation simultaneously, as both occur in the cytoplasm. CRISPR is a method of gene editing that takes advantage of a naturally occurring gene editing system that can be found in bacteria. It uses the Cas9 protein along with a guiding RNA strand that matches the target gene to cut out a specific portion of the gene. After this, when the DNA tries to repair itself, it can cause certain mutations, or it can incorporate a new gene into the DNA sequence.
Gel electrophoresis identifies the sequence of DNA. Gel electrophoresis first uses restriction enzymes to cut the DNA into strands of different lengths. It does this by using dideoxyribose nucleic acid, which is tagged with a fluorescent chemical along with normal nucleotides in order to create segments of DNA that are different sizes. It is able to create every size of DNA possible for a certain gene because it creates an overabundance of copies. The DNA is put into a tube and an electrical current is ran through it. This causes the DNA in the tube, which is filled with a buffer solution, to be sent to the bottom. It detects the sequence as the smallest segments reach the bottom or the positive end first, which are detected by a laser.
In bacterial transformation, plasmids are artificially introduced into a foreign bacteria cell. Plasmids can increase the immunability of bacteria and give them desired traits. Scientists do this by heat shocking the bacteria so the plasmids can enter. An example of a plasmid that scientists can use for bacterial transformation is the P-glow plasmid, which causes an artificial glow to the bacteria.
PCR, or polymerase chain reaction, is a technique used to copy a specific sequence of DNA many times. DNA is denatured in order to split it into 2 separate strands. Primers are then added in the mixture, then attached to the DNA at a specific location, and then start replicating the DNA. The DNA is denatured again, and more primers are used to replicate it once again. This cycle repeats until the specific part of the DNA has been replicated around 1 billion times.
Scientists use recombinant DNA to modify bacteria to produce certain traits. They do this by using plasmids, entering them into the cell by processes like bacteriophages, using restriction enzymes to cut the DNA that was already in the bacteria cell, which lets the plasmid get incorporated into the DNA, ligase, which binds the new DNA to the old DNA, and then it creates a certain trait. A real-world example of this is how biologists have created a bacteria that can produce insulin that humans can use.